everybody. My name is Clara Marchkova, and today I would like to tell you about our research on temporally specific effects of maternal perinatal stress on cortical folding and mood dysregulation in young adulthood. This is research was developed under a collaboration between CAMH, the Center for Addiction and Mental Health, and CETAC, the Central European Institute of Technology. I don't have anything to disclose. Exposure to prenatal stress is a global health problem reported in about 10 to 35% of children worldwide. And it's been shown that it's a risk factor for emotional health problems in the offspring, as well as anxiety and mood dysregulation in young adulthood. However, the mechanisms are not very well understood. Research also shows that exposure to prenatal stress alters the structural and functional development of the brain, and research from our group has shown that mothers exposed to prenatal stress had offspring with more mood dysregulation in young adulthood, who also demonstrated less gray matter volume in the whole brain, and particularly in 3 regions that are known to be hypometabolic among depressed patients versus healthy controls. Those 3 regions included the mid-dorsal frontal cortex, anterior cingulate, and precuneus. However, the gray matter volume develops throughout our lives, and it is a product of surface and cortical thickness. And that's why the current research digs a little bit deeper and looks at gerification, because gerification develops only during the prenatal stage and the first 2 years of life, and represents the folding of the surface area. So we hypothesize that the relationship between early life stress and greater risk of anxiety and mood dysregulation in young adulthood might be actually, at least in part, explained by altered gerification. We've conducted a neuroimaging follow-up of the European Longitudinal Study of Pregnancy and Childhood, which is a prenatal birth cohort born between 1991 and 1992. And these individuals have been followed since the prenatal stage till the age of 20. Thanks to the funding from the Marie Curie Fellowship, we were able to do the neuroimaging follow-up when these individuals were 23 or 24-year-old. And within this particular research I'm presenting you today, we focused on the impact of maternal stress during pregnancy and the first 18 months of life, which was recorded in the early 1990s, on gerification and symptoms of dysregulated mood in young adulthood at the age of 23 or 24. We used FreeSurfer to process the structural magnetic resonance images, and then we calculated the local gerification index as the ratio between the area buried within sulcal folds to the area visible on the outer surface. The dysregulated mood was quantified using the long version of the profile of mood states, which is a questionnaire that includes 65 questions, which are evaluated on a 5-point Likert scale. And it allows us to calculate how much the subject experiences tension, anxiety, depression, dejection, anger, hostility, vigor, activity, fatigue, inertia, and confusion and bewilderment. Maternal stress during pregnancy and the first 18 months of life was measured using the Stressful Life Events Questionnaire, which includes 40 questions answered on a 5-point Likert scale regarding the experience and impact of the life event, This life event could be, for example, death, illness, divorce, job problems, moving, financial difficulties, abuse, etc. And exactly the same questionnaire was administered at 4 different time points. First, mid-pregnancy to cover the stressful life events experienced during first half of pregnancy. Then after birth, to cover the stressful life events experienced during second half of pregnancy. Next, at 6 months of the offspring, to cover the first 6 months. And finally, at 18 months of the offspring, to cover stressful life events experienced by the mother between the age 6 and 18 months of the offspring. And over here, you can see a correlation matrix between these 4 different measures. As you can see, the correlations are relatively high, especially between the early postnatal stress and the prenatal stress measures. However, there's still some unexplained variants, which could be. I would also like to note that while the neuroimaging follow-up was conducted in more than 130 individuals, the analysis I'm presenting today were done only in 85 young adults because not exactly everybody had complete data from all of these 4 questionnaires. The results showed us period-specific effects of prenatal stress on LGI, and we have seen that the timing of the stress influenced location as well as the direction of the effects. We've always modeled the effect of that particular stress period and controlled for stress experienced during the other periods. And as you can see in the table, the VIF and the tolerance intervals suggest there wasn't significant multicollinearity, so that wasn't an issue. What we have seen is that early life prenatal stress showed positive effects on gerification in a large temporal region, which you can see in purple, marked with 1 over here. And we have also seen that later prenatal stress affected gerification, but more in the anterior and smaller regions of the brain, which included also the frontal regions, for example, the caudal middle frontal, which partially overlaps with DLPFC. What we've also seen that while the effects of the later prenatal and early postnatal stress were independent of sex and the effect size was small to medium, the effects that happened during the early prenatal stage, a lot of them were actually sex specific, which I've marked over here with a star. And these effects actually reached medium to large effect size. Interestingly, in females, the effects were positive, which means higher exposure to stress during first half of pregnancy, the greater gerification in several temporal and occipital regions, whereas in males, we have seen negative effects, which showed us that greater exposure to prenatal stress during the first half of pregnancy was associated with less gerification in the frontal regions, parietal and pericalcarine. Follow-up analysis then look at the correlations between the regions we know were related to early prenatal stress and mood disturbance. And these analyses demonstrated that greater mood disturbance in young adulthood was associated with greater gerification in the large cluster in the temporal region. And when we corrected for multiple comparison, this relationship changed only to trend level, and that's why we completed follow-up analysis that didn't overlap between the regions that we know were associated with the early prenatal stress and regions that we found to be associated with mood dysregulation. Those were these 4 regions, which I have marked in the picture in yellow, and the prenatal stress regions you can see here marked in orange. And over here, you can see the middle temporal region, which shows the overlap between the stress and POMS effects. So we are basically confirming what we have seen over here. Since we know, based on past research, that the cortical folding comprises a complex sequence of events that include neurogenesis, cell migration, axon and dendrite growth, as well as generation of synapses. And we also know that each of these processes has its own distinct developmental dynamics and is regulated by growth factors with distinct expression patterns across brain regions and developmental stages. We speculate that the spatial temporal specificity of the effects we observe is likely driven by different cellular or growth mechanisms, with prenatal effects primarily mediated by alteration in neurogenesis and cell migration, and postnatal effects mediated primarily by alterations in neural growth and synapse formation. So overall, we conclude that the exposure of immature brain to maternal stress has long-term implications for cortical morphology and dysregulated mood, and that these findings may have important implications for understanding depression pathophysiology and may open novel opportunities for developing early individualized intervention and prevention strategies. So finally, I would like to thank you for attention, and I would like to thank all my collaborators, particularly researchers from CAMH, Amy Miles and Yulia Nikolova. Amy was involved in the gerification analysis, and Yulia was mentoring the work. And I would also like to thank my colleagues at the Central European Institute of Technology, where we've actually conducted the neuroimaging follow-up of the Ausbach birth cohort thanks to the Marie Curie funding. So thank you.